Okay, you can start whenever.
All right, so we'll talk about what are the potential causes of cancer. This is a very interesting subject. We go back 45 years or so, around 1970, at a time when scientists doubted that viruses cause human cancer. At the time, they knew that viruses cause cancers in animals like Rous sarcoma, Burkitt's lymphoma, but there was no evidence that viruses cause human cancer. 20 years later, I bought a book on microbiology, and it stated that 18% of human cancers are caused by viruses, interestingly. So they don't make definitive statements unless they can actually empirically demonstrate the validity of same. At the same time, let's consider what might cause cancer. You have proto-oncogenes, which are genes that are predisposed to conversion to oncogenes or cancer genes. These are inherited. Then you have environmental factors, chemicals, pollutants in the environment, in the air, the water, the food, and so on. And you have strong electromagnetic radiant sources, power lines, for example, and so on. And when you have structural changes within the genome, then you have the potential for conversion of proto-oncogenes to oncogenes. You also change the structure of the genome. Susumu Tonegawa in the late 70s talked about antibody diversity as a consequence of translocations in genes, deletions, translocations, substitutions, and so on, changes in the genes. And this is critically important. I thought 45 years ago that, of course, viruses could enter the cell. One way would be to utilize their tails as triggers, such that, for example, RNA viruses could inject their RNA into the cytoplasm of cells. The RNA would have a reverse transcriptase enzyme. AIDS works like this to convert the RNA back to DNA. If viral proteins and or nucleic acids find their way to human genomes, they can act as intercalating agents and become inculcated into the genome to trigger increased oncogenic conversions, thus producing cancer cells, because they increase the level of chaos passing through crisis stages, M1, M2, such that you have very shortened telomeres at that point. And what happens in a rarity of cells is that telomerase production is increased, and telomerase is an enzyme that increases the lengths of telomeres. Telomeres are end stops for chromosomes that are critically important in aging. They represent our biological clocks, but they also play a role in cancer. Telomerase is a critically important factor in cancer research today, because if we can stop the production of telomerase in cancer cells, they'll die. At any rate, my point essentially is that viruses play a very critical role in the enhanced expression of cancer.